Hello students, welcome to Read Med Prep Academy channel. Today in Molecular Genetics Part 9, we are going to discuss about regulation of gene expression and Lac Operon model. Chromosomes have packed DNA. DNA is organized into genes. Genes store genetic information. How this information is expressed, this is what we are going to see in this lecture. Here you can see in this image, the cell consists of the nucleus. The nucleus consists of the chromatin material. The chromatin material is made up of the chromosomes. The chromosomes are packed DNA, and DNA are made up of lot of genes. Evidence in support of the idea that genes can be turned on and off is very convincing. Regulation of gene expression has been extensively studied in prokaryotes, especially in E. coli. Gene expression can be controlled or regulated at various stages, at the transcriptional level or the post-transcriptional level, that is the translational level. Here we are going to discuss today about the gene expression at the transcriptional level. In this image you can see that the genes in the DNA are transcripted in the primary transcript, and the primary transcript is made into a messenger RNA, and the messenger RNA goes out from the nucleus into the cytoplasm and gets attached to the ribosomes and causes the translation of proteins depending upon the codon. So the control can be at the transcriptional control, or a RNA processing control, or a RNA transport control, or a translational control. In this we are going to discuss about transcriptional control. Usually small extracellular or intracellular metabolites trigger initiation or inhibition of gene expression. The clusters of genes with related functions are called operons. Here you can see an operon which consists of a control region made up of a promoter and an operator, and next to the control region is the structural genes which transcripts for the messenger RNA, and before the control region is the regulator gene. Operons usually transcribe single messenger RNA molecules. In E. coli, nearly 260 genes are grouped into 75 different operon. Each operon is a unit of gene expression and regulation and consists of one or more structural genes and an adjacent operator gene that controls the transcriptional activity of the structural gene. Here you can see a regulatory region on the left and the operon. Operon consists of a promoter region, an operator region, and the structural genes which help in the transcription of the messenger RNA, and this messenger RNA helps in the translation of the different types of proteins. The structural gene codes for proteins, ribosomal RNA, and the transfer RNA required by the cell. Promoters are the signal sequences in the DNA that initiate the RNA synthesis. RNA polymerase binds to the promoter prior to the initiation of the transcription. The operators are present between the promoters and the structural genes. The repressor protein binds to the operator region of the operon. Here you can see about the synthesis of tryptophan. In the absence of tryptophan, the TRP repressor dissociates from the operator, and the RNA synthesis proceeds and helps in the synthesis of tryptophan. When tryptophan is present in abundant amount, the TRP repressor binds to the operator and the RNA synthesis is blocked. Hence the tryptophan synthesis does not occur. Now let us move on to the Lac Operon model in E. coli. Lac means lactose, that is the genes that control the lactose synthesis. The metabolism of lactose in E. coli requires three types of enzymes. One is permease, two is beta-galactosidase, and three is transacetylase. Here you can see the Lac Operon of E. coli, where it has a regulatory gene Lac-I, the promoter region, and the structural genes Lac-Z, Lac-Y, Lac-A. Lac-Z synthesizes beta-galactosidase, Lac-Y synthesizes lactose permease, and Lac-A synthesizes transacetylase. What are the functions of the enzymes? The enzyme permease is needed for the entry of lactose into the cell. Beta-galactosidase brings about the hydrolysis of lactose to glucose and galactose. Trans-acetylase transfers the acetyl group from acetyl-CoA to beta-galactosidase. What is present in the Lac Operon? The Lac Operon consists of one regulator gene called the I gene, which refers to the inhibitor, the promoter sites called the P site, and the operator site mentioned as O site. Here you can see the Lac Operon. The cap site promotes the RNA polymerase binding. The promoter site helps in the binding of the RNA polymerase for its activity, and the operator site helps in the binding of repressor. If the repressor binds to the operator, there is no activity of RNA polymerase. If the repressor does not bind to the operator, the RNA polymerase activity continues. So repressor determines the activity of the RNA polymerase. In addition, it has three structural genes, LAC-Z, LAC-Y and LAC-K. As already mentioned earlier, LAC-Z gene codes for beta-galactosidase, LAC-Y gene codes for permease, and LAC-A gene codes for trans-acetylase. Here you can see the structural genes of the operon. LAC-Z, which forms beta-galactosidase, it hydrolyzes the lactose into glucose and galactose. LAC-Y gene helps in the formation of permease, which is helpful in the transport of lactose into the cell. LAC-A gene codes for the synthesis of trans-acetylase, which adds acetyl group to the galactose. Who proposed this model? Jacob and Monod proposed the classical model of Lac Operon to explain the gene expression and regulation in E. coli. You can see Francois Jacob and Jacques Monod who discovered the Lac Operon model. In Lac Operon model, a polycistronic structural gene is regulated by a common promoter and regulatory gene. When the cell is using its normal energy source as glucose, the I gene or the inhibitor gene transcribes a repressor messenger RNA, and after its translation, a repressor protein is produced. The repressor protein that is formed binds to the operator region of the operon and prevents the translation. As a result, beta-galactosidase is not produced. Here you can see when the lactose is absent, the repressor is active and operon is off. That means when the lactose is absent, the regulatory gene produces the messenger RNA that forms the repressor protein, and this repressor protein binds to the operator and prevents the activity of the RNA polymerase. In the absence of the preferred carbon source such as glucose, if lactose is available as energy source for the bacteria, then lactose enters the cell as a result of permease enzyme. Lactose acts as an inducer and interacts with the repressor to inactivate the repressor. Normally, the repressor protein binds to the operator of the operon and prevents RNA polymerase from transcribing the operon. Here you can see the activity of the various enzymes. Permease actively transports lactose into the cell when lactose is necessary, and the enzyme beta-galactose breaks the lactose into galactose and glucose. And beta-galactose also converts the lactose into the related compound allolactose. The allolactose acts as an inducer. Allolactose is also converted into galactose and glucose. Inactivation of the repressor occurs when there is presence of inducer, such as lactose or allolactose, which are inducers. The repressor is inactivated by the interaction with the inducer. Here you can see when lactose is present, the repressor becomes inactive and operon is on. So when the lactose is present, there is formation of lactose and allolactose. Allolactose acts as an inducer. It binds with the repressor, and the repressor does not get attached to the operator. Hence, the operator is free and the RNA polymerase activity continues and helps or stimulates the synthesis of the messenger RNA for the translation of the enzymes beta-galactose, permease and trans-acetylase. This allows the RNA polymerase to bind to the promoter site, and the operon produce Lac messenger RNA. It enables the formation of all the enzymes, and these enzymes are needed for the metabolism of lactose. Here you can see, in the absence of the inducer, the repressor binds to the operator region and prevents the RNA polymerase from transcribing the operon. In the presence of the inducer, the operator stimulates the binding of the RNA polymerase and the repressor becomes inactive, and the RNA polymerase helps in the synthesis of Lac messenger RNA, and this helps the translation of the enzymes. The control of Lac Operon, how is it done? By a negative control. It is also having a positive regulation as well. To understand it, we can see on the left side the positive regulation. When the activator binds with the operator, the transcription continues. When the activator does not bind with the operator, no transcription occurs. The negative regulation, when the repressor binds with the operator, there is no activity of RNA polymerase, so no transcription. If the repressor does not bind with the operator, the RNA polymerase activity continues and transcription of the messenger RNA occurs. So today we discussed in molecular genetics part 9 about the regulation of gene expression and the Lac Operon model in E.coli. Thank you. Kindly subscribe, like, share and comment to channel Read MedPrep Academy. Log on to www.readmedprepacademy.com. Our Facebook ID is Read MedPrep Academy. Our email is readmedprepacademy@gmail.com. Kindly post your questions in the comment box. We will reply with appropriate answers. Thank you very much.